Okay, so good evening, everybody. I'm going to start again. So last week we started talking about control of gene expression, how gene expression is controlled in different types of cells, but we focused mostly on control at the transcription level, transcription where the DNA is transcribed into messenger RNA, right? And there are various mechanisms that we discussed that are used in different types of cells to control the transcription. So for example, in bacterial cells, we talked about constitutive and inducible promoters and different types of operons in bacteria. There are some negatively regulated operons and positively regulated operons, things like that. All of that focuses on whether the gene is transcribed or not and what controls it, right? And then in eukaryotic cells also we talked about how transcription is regulated first at the level of packing, DNA packing, where the access could be limited, and secondly through the use of different control elements, including of course the promoter region and other regions like enhancers, where activators bind and different transcription factors come into play to control the expression of genes in eukaryotic cells. So that is where most of the control is exercised, but there are many other control mechanisms that exist in eukaryotic cells. And so today we are going to be looking at other mechanisms of controlling gene expression other than the transcriptional control that we talked about last week. Okay. So first of all, after transcription, let's say that you have the gene. Here is your gene, and let's say that you are able to transcribe. So you have overcome the first control point, and so now you have from the gene you have a messenger RNA coming out. So this is the messenger RNA. And this is the gene. Now there are various ways in which the RNA is processed. If you remember when we talked about transcription and translation, we talked about RNA splicing, right? Where the first transcript that you get off of the gene is longer than the mature messenger RNA because it has those introns in it, right? So there is RNA processing going on. So for example, you have addition of the cap and tail. Addition of the cap and tail. So there is a on the messenger RNA on the 5' end there is a cap put on, and on the 3' end a long tail that is added. And that we'll talk about the functions of those in control of gene expression. And then messenger RNA, when it comes out of the mature RNA, when it comes out, it depends on how stable it is. How long does it stay in the cytoplasm? That can also control the expression of gene. And finally, how the RNA is spliced, because there are various ways in which it could be spliced. So we'll talk about each of this next. So RNA processing can control gene expression to some degree. So to recall what we had learned before, here is our gene with its exons and its introns. So remember, exons are portions of the gene that are actually expressed. So those are coding regions. And introns are intervening sequences that are not expressed. So that's kind of junk DNA, right? So here is the mature, the primary transcript. This is the primary transcript that has the introns in it. It has the introns in it, right? And then using certain enzymes and other proteins, you can the cell excises out the introns and joins together the exons to make the mature messenger RNA. And you also get this poly A tail and a cap on this is the 5' end, and this is the 3' end. Okay, so you get a cap and a tail added to the messenger RNA. Okay, so let's look at it a little bit further. What does the cap do? Cap for the messenger RNA? Well, it stabilizes the messenger RNA, and it is involved in messenger RNA splicing. So, you know, the way the cell excises out the introns, the cap is involved in interaction with the molecules that come along to cut out the introns. And it is also involved in export of the messenger RNA from the nucleus, okay, and recognition for translation. So when the messenger RNA gets to the ribosome, the cap is involved in recognizing the place where the messenger RNA is going to attach itself to the ribosome, the large ribosome subunit, right? So those are all the functions of the cap of the cap of the messenger RNA. And the poly A tail is nothing but a long tail of there is a consensus sequence, but a lot of A's in there. There is also a U. So the book has a consensus sequence in it, but essentially that long tail, it stabilizes the messenger RNA because it adds more stuff to it, and it partly determines the messenger RNA half-life. What do we mean by half-life? How long the messenger RNA is going to stay in the cytoplasm. So what happens is when you have this messenger RNA and it has the cap and the tail, the cap and the long tail right here. My yellow color is not that good. Can you see it? Okay. So this tail. So imagine there is a dog trying to, cut out the tail. Slowly chew at the tail. Okay. So basically, it's lopping pieces off of the tail and trying to get to the messenger RNA. So so over the length of the messenger RNA, the tail keeps getting shorter and shorter and shorter as the messenger RNA ages, right? And so at some point it becomes so short that this becomes now vulnerable to degradation, and it in fact degrades, right? So the length of this tail will have some impact on how long the messenger RNA is going to stay in the cytoplasm. And the longer amount of time it stays, the more protein it can make by translation, right? And the shorter amount, if it is not so stable and the half-life is short, then you will make less amount of protein. Okay, so that's why poly A tail is involved in it determines half-life of the messenger RNA. Okay, now let's talk about alternative RNA splicing. So what I had told before is that the RNA is spliced to get rid of the introns and splice the exons together, right? Well, it could be a little more, for some proteins, it could be a little more complex than that. So for example, here is an example of here is a gene with exons 1, 2, 3, 4, and 5. Okay. Now, when this, and here is the primary transcript. But when the mature transcript is formed, what can happen is sometimes um, you could get rid of this whole segment. So the two introns as well as the exons. And if you do that, then you will end up with this transcript. On the other hand, you could excise out this segment. And if you excise out that segment, then you will end up with this other alternative transcript. So starting with the same gene, you can get two different messenger RNA transcripts. Now, the messenger RNA transcript is what determines what protein is being made, right? Because this has a sequence of amino acids that's going to be utilized. This has the codons for the amino acids. So obviously, this protein is going to look a little bit different than this. The protein that is made with this transcript is going to look a little bit different than the protein that is made with this transcript. So starting with the same gene, you could make two different proteins, right? And so depending on what kinds of enzymes come into play over here, you could go either one way or the other. Okay, so this mechanism of gene control of gene expression is called alternative RNA splicing. So we have so far learned that every gene codes for one protein, right? One gene, one protein. Well, it turns out that sometimes one gene can code for multiple proteins, and this is the reason why, when people looked at the human genome, because we have lots and lots of proteins in our body, they were expecting to find close to 100,000 genes, But when they actually sequenced the human genome, what they found was more like 20 to 30,000 genes, more like 21,000 genes. So that was a surprise, and one of the explanations why there are so much fewer genes than what we expected is that one gene could be coding for multiple proteins. So even though we have only 21,000 genes, we have a lot more proteins than that. So the old hypothesis that one gene, dogma of one gene, one protein is no longer true. So are we good with alternative RNA splicing, how that can happen? It happens because sometimes exons that are flanked by two introns, they just get spliced out. They can get spliced out. Okay, so that is one mechanism of how genes can be controlled after transcription. Now, another mechanism is, I think I had posted a question about this in the discussion forum, is interference RNA. What do we mean by interference RNA, RNAi? Well, interference RNA are molecules, RNA molecules that are short. So they are just a short sequence of nucleotides, RNA nucleotides, that are complementary to the messenger, to some particular part of the messenger RNA. So here is the messenger RNA, and so here is a this is the interference RNA that will bind. So here is the RNAi that will bind to the your messenger RNA, and then one of two things can happen. It can either break this messenger RNA down, degrade it, or it can block translation, because the messenger RNA then cannot attach to the ribosome, right? So these things are also called microRNAs, micro interference RNAs. And they are single-stranded chains of about 20 nucleotides long. One side of it binds to a protein complex, and the other side binds to the that's complementary to the messenger RNA to interfere with protein production. So let's take a look at that. So here is the protein. Here is the microRNA, micro interference RNA, right? And so the protein attaches to the microRNA right here, forms a complex, and then it attaches to the target messenger RNA right there with hydrogen bonds. And once that happens, it can either destroy the messenger RNA, degrade it, or it can block the translation because this is blocking it from going through the ribosome. Okay, so again, presence or absence of these microRNA, interference RNA, will lead to more or less production of the protein that the gene codes for, right? So that's another level of control that the cells can exercise in controlling the gene expression. How is everybody doing? I need to know that you are understanding things. Okay, thanks. Okay, so next, you can also get gene expression by other mechanisms, control of gene expression by other mechanisms. So, for example, breakdown of messenger RNA. We talked about this, especially with the poly-A tail, right? Poly-A tail is controls the half-life of the messenger RNA, and it turns out that some messenger RNAs hang around for a long time, right? So some messenger RNAs could hang around for hours to even days, whereas others they have half-life in seconds. So obviously, the proteins that are not needed in large quantities, the half-life of those messenger RNAs can be really small. So as soon as the protein is made to the right amount, the messenger RNA degrades. On the other hand, if you need a lot of the protein, then obviously you want a much longer half-life for those proteins. Okay, so breakdown of messenger RNA can be used to control protein expression. Initiation of translation, you can control protein expression by blocking initiation of translation. This is a little bit different than what I just talked about, the micro interference RNA. That's one possible, but you can also block translation by other mechanisms. So that's another way of controlling gene expression. Well, sometimes you go through the translation process and you actually make the protein. You actually make the protein, but the protein may not be active, and it may need a particular enzyme to actually activate it. Okay, so one example of that is insulin. We will talk about that in a minute, and I'll show you how that works. So protein activation, you need another enzyme to activate the protein. And finally, even after you have made protein and it is active, you could still exercise control over how much protein you have by having things break down. So if the protein is subject to degradation within the cell, then it may be active only for a little time and then it breaks down. So that's another level of control that the cell exercises. So I gave you an example of insulin. So for example, this This here is the active form of insulin. And you notice that there are two polypeptide chains that are stabilized by these disulfide linkages, disulfide bonds, right? So there are two, but when the initially insulin is made, it is one polypeptide, this one whole polypeptide, right? But then an enzyme comes in and clips the protein in these two spots. And so then it makes two polypeptide chains, and they form these disulfide bonds. Um, first the disulfide bonds are formed, then it is clipped so that you have two polypeptide chains stabilized by the bonds. And this is the active form of insulin. So unless you have the right enzyme to clip the protein, this active form is not going to happen. So in some proteins, this kind of activation is necessary for activation of the protein, right? So that was another thing. So let's kind of go through all the levels of controls that we have so far talked about. Okay. For example, we started off. You always think about starting at the main source of gene expression. The main source is, of course, DNA. And if you have DNA that is tightly packed, so DNA packing or unpacking, if it is tightly packed, then you won't have gene expression unless it opens up and is accessible to the RNA polymerase, right? And then the second, so this is the first level of control. The second level of control was what we discussed last week, control of transcription. So all the regulatory proteins and regulatory control sequences, promoters, enhancers, operons, things like that, that's control of transcription. That's the second level. Then comes control of RNA processing. So the addition of 5' and 3', 5' cap and 3' tail, and alternative splicing, alternative RNA splicing. These come next in the flow of control of gene expression, right? Then of course we didn't talk about this, but remember the nucleus has pores in it, the nuclear pores, and so here is the nucleus, and messenger RNA has to come out through the pores to the cytoplasm to get to the ribosome. So that also can control the amount of messenger RNA or the rate at which the messenger RNA comes out, the pores, right? So flow through the nuclear envelope is another level of control. Then we talked about breakdown of messenger RNA depending on the poly A tail. And we talked about control of translation. Here we also talked about micro interference RNA. And then control after translation, which was either cleavage, modification, or activation of the proteins, and then breakdown of the proteins. So these are also, so you can see how complex the control mechanisms are in cells, and we call it a cascade of controls, right? So there's a so if you can imagine, think about when you open a tap in your house to get some water. Does the whole so the water is coming from a reservoir somewhere in Northern California, right? Lake Shasta or somewhere like that. And so you can imagine here is the big lake. And from the lake you have some, control pumps and valves and all kinds of things going on. You have the river, the canal coming down to the Southern California, then it goes to a reservoir near your home. And then from there it has more pumps and valves, and by the time it reaches your home, the tap, you only have a little amount of water. You can literally control the amount of water you get starting from the reservoir. If the whole reservoir were to empty in your house, that wouldn't do you much good, right? The same is true with genes. You don't want all 21,000 genes being active all the time. Otherwise, your body will be out of control, right? So this is the same mechanism. Show you in a here's the so think about the analogy. This is your reservoir, and you have a whole set of controls by the time you get water in your tap. Same thing over here. Here is your reservoir of genes that you have plenty, lots and lots of genes to make lots and lots of proteins, but you control the expression to get just the right amount and the right kinds of proteins. Okay, and what we discussed right now, what we have been discussing is at various stages you can exercise control. Okay, so that's the analogy for gene expression. So let's see whether you can apply your knowledge to these situations. For each of the following, determine whether an increase or decrease in the amount of gene product is expected. So the messenger RNA fails to receive a poly-A tail during processing in the nucleus. What do you expect? Do you expect to see more protein or less protein? Okay, Victoria says less because it is not stable, right? It's not stable, so it will break down faster. So you will get less protein. The messenger RNA becomes more stable and lasts twice as long in the cell cytoplasm. So obviously, in this case, you're going to get more. Yeah. Okay, and then how about this? The region of the chromatin containing the gene becomes tightly compacted. The region of the chromatin. Chromatin is the DNA and protein complex in the nucleus. Chromatin is DNA and protein complex in the nucleus. Yeah, that's right. There is no unpacking because it's more compacted, right? Yeah. So then it should become less. So you should get less amount of protein if you can't access the DNA, right? So you will get less product. Okay. An enzyme that is required to cleave and activate the protein product is missing. What's going to happen here? Let me see. Richard, Jerry, Kathy. Anybody? Okay, this is going to be a good quiz question then. Is everybody awake? Okay, Jerry is awake. So when an enzyme is missing, the enzyme that activates the protein, then obviously you're not going to get active protein, right? So you will get less product, less gene product in this case. So these are just examples of how things can be controlled. Okay. and talked about how how gene expression. Now, although we talked about all these levels of control, I want to emphasize that we still don't know all the mechanisms of control of gene expression. So this is an area of research that's ongoing. So we know some amount, but still an ongoing research, right? Um, because it's so complex, the protein production is so complex. There are so many different enzymes involved, so many different molecules involved that it's going to take a long time before we figure everything out. Okay, now there are other control mechanisms as well. So we'll talk briefly about those. So for example, I don't know if you've heard of a woman, her name was Barbara McClintock. Barbara McClintock, she got the Nobel Prize in, I don't know which subject, but she got the Nobel Prize for her research. And her research was basically on these things called transposable elements. Transposable elements. So what these are, transposable elements are elements of DNA that can actually jump from, so let's say that you have an element in one gene right here. And here is another chromosome. So this blue element can jump from one chromosome to another chromosome. Okay, so these they used to call them jumping genes, right? They are not really genes, but elements of DNA that literally can transpose from one chromosome to another chromosome. And sometimes when these elements jump or transpose, what they do is they insert themselves inside the locus of a gene, right? So, for example, the corn, the Indian corn that has many different colors, it's because of these transposable elements. What happens is the one element will jump into the gene for a particular color and disrupt it. And when that happens, that color stops being expressed. And then that element will move on somewhere else, and this gene now starts expressing again. So you get all kinds of patterns of color patterns because of these transposable elements. They are just jumping from one chromosome to another, randomly cutting into different genes and stopping their expression for some time, and then they will move on again. Does that make sense? This is because of the transposable elements. Okay. Then there are gene rearrangements. So there are ways that genes can rearrange themselves. And when they rearrange themselves, they start to make different types of proteins. So imagine that you have a particular gene, and that gene has the sequence of a particular, a sequence of amino acids. But if I were to take a section from here and rearrange it over here, what's going to happen? The sequence of amino acids is going to change, right? And so you're going to get a different type of protein. And this is particularly important in antibody diversity. You know that when we get an infection, we get a foreign particle, foreign molecule in our body mounts an antibody response, right? It can produce antibodies that are specific, that will grab onto the invading foreign particle and destroy it, right? How does it do that? how can it respond very quickly to foreign particles, foreign molecules coming into our body? And the way it does that is by there are a certain number of genes that can be rearranged very quickly. And by rearranging them, arranging those genes, the body can produce molecules, these antibodies, that have a lot of different shapes. So there is an infinite variety of shapes that the body can produce just by rearranging certain elements in these genes. Okay, think about just by changing Lego pieces, you can make different shapes, right? Same thing with same thing with the body, with the genes for antibodies. There are certain elements that it will rearrange to make proteins that fit different shapes, and so we can mount a response to any invading proteins or antigens. So gene rearrangement is another way that we control what kinds of proteins get produced. Okay. Then you have tissue-specific regulation. So, for example, there are certain tissues that will produce only certain proteins. For example, hemoglobin. Hemoglobin, where is it produced? Only in the red blood cells, right? Only in red blood cells. So there are, people have found out there are regulatory sequences in the gene for hemoglobin that somehow control the expression of those genes only in certain types of tissues, right? So tissue-specific regulation is another way of controlling gene expression. Okay? And then coordinate expression of genes. What do we mean by that? So let's take a look at that. What do you mean? What's a cell cycle? Well, every cell goes through a certain cycle. Okay, so every cell has to divide, right? Every cell has to divide, and the way it divides is it goes through a cell cycle. So let's say that here is a new cell. Right here is a new cell, and in order to divide, the cell has to first grow. It has to double in its size, right, before it divides. Otherwise, you will get smaller and smaller and smaller cells. So what it goes through several phases. This whole thing is a cell cycle. Okay, this whole thing is a cell cycle, but it goes through several phases. So here is the first phase. This is called it's called the G1 phase. G1 phase. Okay, and during the G1 phase, what's happening is the cell is basically growing. It's making more material so that it increases in volume. Okay, and then the second phase is the S phase. This is the S phase right here. And during S phase, the DNA replicates. So DNA is synthesized. So that's why we call it the S phase, DNA synthesis. And then the third phase is G2 phase. And here it is getting ready to divide. So it's making a lot of proteins that are necessary for the division, right? Cell division. And so finally, this is the mitotic phase. Mitotic phase is when the cell actually divides into two. So at this point, you get two cells. So starting with one cell over here, after the cell goes through the cell cycle, you get two cells. And then each of those new daughter cells will go through the same cell cycle all over again, right? To divide and divide again. But we know that the cells don't keep dividing all the time, right? So there has to be control of cell division. And the way it is done is, so you know how your dishwasher goes through the whole dishwasher cycle or the washing machine cycle is divided into different parts. So first you have the water coming in, then you have the spinning, and then you have the soap there are all these different parts of the washing machine cycle. So this is kind of similar to that. Now, so there are different checkpoints. So here is something called the G1 checkpoint. Here is something called the G2 checkpoint. And here is something called the M checkpoint, where it goes into the mitotic phase, right? So at each checkpoint, certain molecules have to be present, certain signals have to be present, okay? Without the presence of these signals, the cell will not proceed to the next phase, okay? So it's just like a stoplight. So if the stoplight is off, then your cell is going to go into a quiescent phase called G0 phase. So this is a quiescent phase. So the cell can spend a lot of time in G0 phase until some signals come in and say, okay, now it's time to divide. So those proteins tell the cell, the DNA to start dividing. And so the DNA starts to divide and then comes over here, and there is another control point and says, wait, we need all the presence of all these proteins in order to move forward. So you can see that there is an exquisite cascade of control in cell division. And so you have to have, so this is called coordinated control, right? So all these proteins have to be expressed around the same time. So and when they are expressed like that, then the cell cycle moves forward. Okay, so this is coordinated control of gene expression. Okay, so now you might ask that sometimes the cell gets a signal from outside. The cell gets a signal from outside to say, you need to start dividing. Okay, so there are many growth factors that signal the cell how to start dividing. So how does that happen? How can a molecule outside the cell actually make things happen inside the cell without even entering the cell? Okay, so let's take a look at that. So for example, here is a growth factor, which could be a protein, or it need not be a protein. It can be a small molecule. Okay, but the important thing is, so here is the cell membrane right here, cell membrane, and you know that the cell membrane has lots of proteins embedded in it. So one of these proteins might have a receptor for this growth factor. Okay, so what happens is when this growth factor comes and binds to the receptor on the cell surface, conformational changes happen to the protein inside, and these conformational changes cascade into a whole pathway that will induce production of certain proteins. So without this signal here, these proteins are not going to be produced. Okay, and it all takes place because of a conformational change right here in the receptor protein, right? So you can imagine that there is a lot of research going on in receptor proteins. Receptor proteins are proteins that are embedded in the cell membrane on the cell surface, and they get signals from outside to control production of different proteins inside. Okay, so then if a growth factor comes in and produces certain proteins, it will signal the cell cycle to go past the G1 and so on and so forth. Okay, so this is called signal transduction pathway. All right here. So a signal from outside the cell comes in and makes things happen inside the cell. My analogy to this is if you go to a fast food restaurant, okay, uh, so you can imagine that you are this person going to the fast food restaurant, and all you do is you go to the cashier. You go to the cashier, right? And you can order a hamburger. Do you go inside the kitchen and cook the hamburger yourself? No. So you are essentially a signal molecule that gives a signal over here at the cashier, and all of a sudden, signals go to the cook to the cook inside that produces the hamburger, and the hamburger comes out, right? And you never went inside the kitchen, but because of the signals that you gave, things happened inside the kitchen, and something came out. The same is true with protein expression control, right? You can have a signal outside, and it can literally make things happen inside the cell. In fact, it can make a whole lot of things happen inside the cell. So a whole lot of signals can be transduced, and proteins can be produced. Does that make sense? Okay. So we talked about um, signal transduction and how that controls expression of proteins inside the cell. Now, our body is quite complex, and especially when we are growing from a baby stage, actually from an embryo stage, developing into a whole body. You can imagine that there has to be a lot of control that is exercised, because how does the embryo know where to put the head and where to put our legs? So here is the embryo. How does it develop into this side is going to become the head, this side is going to become the legs? There has to be some kind of control being exercised so that the appropriate genes are expressed in one part and the other genes are expressed in another part, right? So there is a whole set of control genes, and these are called homeotic genes. Homeotic genes are master control genes. These are the set of master control genes that determine the anatomy of the body, specifying structures, how things are going to develop. So I'm going to give you just one example of how homeotic genes can exercise control over that. Okay, so here is an example of how people figured out that there are these master control genes. Here is a fruit fly. Normally, the fruit fly has these two eyes. So here is one eye, and here is another eye, and the eye has these antennas, right? Well, they did a mutation in the homeotic gene, then the developmental gene in the fruit fly, and guess what happened? Instead of having the antenna, they had legs coming out of their eyes, right? So, oh, no audio? Okay, Victoria says she has audio. Richard, can you hear me? No. How about Kathy and Jerry? Okay, so Kathy can hear me now, but okay. You need to let me know if you can't hear me. Okay? So, Richard, problem at your end. Okay. I want to make sure, Jerry, can you hear me? Oh, okay. So, all right. So we're almost done. So I'll wait a little bit. Okay. So, we are talking about the homeotic genes, which are master control genes that will express proteins, certain proteins at certain times, and that allows for development of the different parts of the body, right? So let's take an example of how that happens. So here is a egg cell. Here is an egg cell, and the egg cell has and this is the fertilized egg, right? Fertilized egg. Now the egg cell has certain nerve cells around it, surrounding it. Okay, and what these nerve cells do is they send in a messenger RNA for a protein called the bicoid protein. Okay, so remember, this is just messenger RNA right now. So if the messenger RNA is going to be produced this way, then what happens is the messenger RNA is going to diffuse through the egg cell, and you're going to have higher concentration of the messenger RNA over here and lower concentration over here. Okay, and after the egg is fertilized, the messenger RNA is translated into the bicoid protein. And so it turns out that you get more bicoid protein on this end and less bicoid protein on this end. And that's what determines the placement of the head and the tail. This is for a fruit fly, by the way, for a fruit fly. Okay. So you can see how the diffusion of the messenger RNA is actually controlling the expression of protein, because there is more messenger RNA here than here, and so that determines the axis of the body. You have head on one side and the tail on the other side. Okay, so this is just one example of how the body exercises control over gene expression in the development of the body as the body develops. Okay, so pretty complex. You can imagine that this is just one part of development, right? There are lots and lots of development activities going on all over the body. And so there are similar controls in place for gene expression. Okay, so let's talk about some review questions. So what are the cap and poly A tail structures in messenger RNA, and what are their functions? Okay, so Victoria says 5' and 3' end. So the cap structure is at the 5' end, and the tail structure is at the 3' end. And what is a poly A tail? It's basically a long chain of A's, mostly A's, but there are there are other consensus sequences in there. It's a long chain of that A nucleotides, and it determines the half-life. Correct? Yes. Okay. Now, how do interference RNAs, iRNAs regulate gene expression? Somebody other than Victoria. Richard, Kathy, Jerry. How does the iRNA control gene expression? Okay, let's start with what is iRNA? Yeah, interference RNA. And what does it look like? Okay, Richard is writing something. Okay, it prevents expression. How does it do that? It has a certain nucleotide sequence, right? It has a certain nucleotide sequence that is complementary to the messenger RNA, and so it will bind to the messenger RNA, and it could do one of two things. It could destroy the messenger RNA, or it could block the translation, right? Okay. Can you give an example of regulation of expression at the initiation of translation? Initiation of translation. So remember, you have the ribosome, and the messenger RNA comes in here and attaches itself to the ribosome. And something could block the initiation. You could have either interference RNA come and bind to it and block the translation, or you could have another enzyme come in and block the translation. Okay. Can expression of genes be regulated after protein has been synthesized? After the protein has been synthesized, can it be regulated? Boy, I have a quiet group today. Can you hear me? Oh, I know. Okay, Kathy is responding. Oh, Kathy had issues with the computer. Oh, sorry about that. Okay. Yeah, so Victoria is saying you can have cleavage of the protein to activate it, right? You can activate by cleaving the protein at certain points. That's one way. And another way is you could break down the protein. You could have enzymes that break down the protein. Okay. What is alternative RNA splicing, and how does it control gene expression? Alternative RNA splicing. So remember the introns and the exons. So these are the introns, let's say, and the exons. So you could have, you know, one exon not be included or another exon not be included. So you could get two different transcripts from the same gene, and therefore you could get two different proteins with the same gene. Okay. And how can molecules outside the cell control its gene expression? So this would be the growth factor, right? Growth factor giving a signal, and the signal being transduced. So this is called signal transduction. Okay. Okay. So well, this was a pretty quiet session. I guess some people had issues hearing. Richard, Richard, are you able to hear me now? I hope you didn't have loss of audio for too long. Okay. Well, it is what it is, and I guess you can always go back to the archive to listen to it. Or feel free to ask me questions during the lab next week. So, what do you have questions today about today's lecture? Okay, question is on what will be the quiz on Monday. So the quiz on Monday will be on control of gene expression, both lectures, right? So today's lecture and last week's lecture. Okay, so Victoria is asking whether microRNA is the same as microRNA. As long as it interferes with the RNA, messenger RNA, yeah, it is the same micro interference RNA. I guess you could have microRNA sequence that, uh, yeah, it is microRNA. Okay. Okay, so Kathy is asking whether the growth factor that I spoke about is another sequence of nucleotides. Not really. So the growth factors are not, it's not a nucleic acid, it's not DNA or RNA, right? So think about it this way. Here is my cell, and here is the nucleus, and the DNA is inside here, okay? And there is a protein on the cell surface which has a receptor. It has a receptor for a particular growth factor. So now this growth factor that comes in can be anything, can be a small molecule, or it can be a protein. It's a lot of times it is a protein, okay? And so the protein will come in and it has a certain shape that fits right there, okay? It fits right there. And when it binds to the receptor, it makes a whole lot of changes happen that tells the DNA over here to start producing a protein that will allow the cell to grow. That will signal the cell to start growing. So this growth factor is not a nucleotide. It's not a sequence of nucleotide. It may be either a small molecule or a protein that binds to the receptor protein over here in the cell membrane. And because of that binding, a whole set of changes occur that tells the DNA, the gene inside, to produce the right kind of protein to make the cell grow. Okay, and the next question is the source of binding specific to a particular cell? Yeah, because the cell, so that's a good question. Different cells will have different receptors on their cell surface. Okay, so for example, you might have heard of the HER2 receptor. Have you heard of the HER2 receptor? It is one of the receptors that is expressed a lot, overexpressed on the certain kinds of breast cancer cells. Okay, and so because it is overexpressed on these breast cancer cells, it will the cell grows too fast, and it that's why uh, some women will get breast cancer. And so they are said to be HER2 positive. That means their breast cancer cells are overexpressing this particular protein. Okay, so different cells are going to have different cell surface proteins and different receptors on their cell surface. And again, that is also controlled at some level by the genes being regulated, right? So it's a very complex set of regulation. So Richard is saying, is it kind of related to the cell division? Yeah, it is. Okay, and Richard is saying he's having problem with the audio. Okay, I'm sorry. So Kathy is saying if a cell is susceptible to mutation, that could be challenging. Susceptible to mutation, what do you mean by that? Cells have the DNA inside their nucleus, and the mutations can happen in a variety of ways. Mutations can be induced by radiation, or they can be induced by chemical agents, or they could happen randomly. Okay. Here you said last week you mentioned HIV mutations. Oh, okay. So HIV is a virus, right? And the virus, these viruses do mutate quite often, right? Our cells don't mutate that often. Viruses, because they are, they only have a protein coat on them. They can mutate quite often, especially RNA viruses. Viruses that have RNA as their genetic material, they mutate more often than DNA viruses. Okay, so remember, viruses have DNA or RNA inside, and they are covered by a protein coat. They're covered by a coat of protein. So viruses don't have all the stuff that we have. They don't have a nucleus, for example. So a protein coat, and so this is the protein, and some sort of genetic material inside. So there are viruses that have DNA inside, or some of them have RNA inside. So you can have DNA viruses or RNA viruses. And especially the RNA viruses, they mutate a lot. Okay. But our DNA doesn't mutate that often unless we are exposed to, mutagens like radiation or tobacco smoke, things like that can cause mutations. Okay. All right. Other questions? Okay. So I'm sorry that we had problems with audio today. Hope that doesn't occur next time. And as I said, you can always go back and look at the archive, or ask me questions in the lab, during the lab. and I'll be happy to answer them. Okay. So I will see you next Monday in the lab, and have a good weekend. You're welcome. So I'm going to stop recording now.